review a little bit about bulk flow, because essentially, we talked about some of the functions of the circulatory system. It's a transportation mechanism. It's how we transport nutrients and wastes, electrolytes, hormones throughout the body. We all know this. So the thing is, we got to get stuff that's in our bloodstream into our tissues, and we got to get stuff that's in our tissues back into the bloodstream so we can move it to the appropriate place. So bulk flow is going to help facilitate this. And so we've already been introduced to gradients of some sort. Back in 210, you learned about concentration gradients, moving a solute or a substance from an area of high concentration to low concentration. Chapter 19, we talked about pressure gradients, how we move, a fluid, i.e. blood, from an area of high pressure to low pressure. So we're going to facilitate that same kind of concept here when we talk about bulk flow in our bloodstream here. So again, we're going to be moving material down a pressure gradient. So keep that in mind. So we're going to move that material from an area of high pressure to low pressure. And so, of course, when we discussed this in the previous class, these pressures will, in some cases, be at opposition with one another. So we learned about the different types of pressure, hydrostatic pressure, and then colloid-oncotic pressure. And so those pressures are in opposition with one another. And it's just like I said with last class, to tug of war. Whoever is stronger, whoever has more pressure is going to win out. And so wherever we see the higher pressure, that's where we're going to see the movement of material from that area of high pressure to low pressure here. So the two pressures, hydrostatic versus our colloid-oncotic pressure there. And so that brings us to the two concepts of what we call filtration and resorption. And we'll visit this again in chapter 24 when we talk about the urinary system, right? But filtration, and make sure that you know this, and we talk about fluid, it's going to move out of the blood. So out of the capillary into the tissues, okay? Out of the capillaries into the tissues. And so again, it depends on what type of capillary we're talking about. We had the three different types of capillaries, the continuous, right, the fenestrated, and the sinusoidal. And continuous is the least leaky. And then we move to the fenestrated, which has some holes in the cells, all right? So more fluid can leak out of that. And then we have the sinusoidal, which you have these big gaps, right? So as we progress through those capillaries become more leaky. And so more fluid, but also, right, certain types of solutes can leave. For example, we don't want blood cells, blood cells, which would be white blood cells and red blood cells. We don't want them leaking out, right? So the capillaries will maintain the larger substances, right? Like the foreign blood elements. Mostly the large proteins too, because, and I'll review it here in a second, right? We don't want proteins leaving our bloodstream because proteins leave the bloodstream and go into the tissue, and that affects the oncotic pressure there. And so that can cause tissue retention in fluid retention in our tissues. And we don't want to mess around with that, all right? So keep that in mind. So when we're dealing with filtration here, right, we're going to move things out of the blood, and hopefully we're going to be blocking those large solutes. So when we get into our capillary bed, this guy here, right, that's what we're going to see on the arterial end. We're going to see primarily filtration. So you're going to see, look at the arrows, look what they're showing us, fluid moving out of the blood vessel. And that's what we want to see on the arterial end, which makes sense because we might want to get some glucose, we want to get some respiratory gases, we want to get nutrients to leave the bloodstream to go out to the tissues there. All right, so that is going to be filtration. All right, so resorption is we're going to move, substances, fluid back into the blood. And so we primarily see that at the other end of our capillary bed, which is the venous end, which makes sense because we want to get cellular waste, carbon dioxide to leave the tissues because they're undergoing their metabolism. And we want to dump that material back into the bloodstream and get it out of there and send it back to the appropriate location, i.e. carbon dioxide needs to go to the lungs so you can exhale it out. Right, so this is kind of what we want to see when we're discussing our bulk flow here, right? So we're talking about those two concepts, filtration and resorption. So back to hydrostatic pressure now, all right? So we're going to now talk about, right, the two different types of pressures, hydrostatic versus colloid oncotic pressure. So what is hydrostatic pressure? So that is the force that is exerted on the vessel walls, right, by the fluid. So like water in your pipe of your house, all right, hydrostatic pressure, it presses on the walls of the pipe. Your blood and the fluid there, all right, is going to press on the vessel walls there. So we have two types of hydrostatic pressure. You have blood hydrostatic pressure, and you can see how it's, all right, noted here, HP, and then subscript B. And then we have interstitial fluid hydrostatic pressure, HP subscript IF. Right, so that's how we're going to be able to differentiate between the two. Now, the hydrostatic pressure, the blood hydrostatic pressure, should be the larger of the two. Right, and in fact, interstitial fluid hydrostatic pressure should be pretty much zero. Right, so essentially, that's the force that's pressing on the vessel wall from the outside. So the blood hydrostatic pressure is going to be the pressure that the fluid in your blood vessel is putting on the inside of the blood vessel wall. It's trying to get out. So therefore, it is going to promote filtration. Because what did we say? Filtration, just to review, right? Filtration is fluid moves out of the blood. Okay, moves out. So that's what we're looking at here. The blood hydrostatic pressure is going to be trying to get out of the blood vessel, and so that is going to promote filtration. Interstitial is going to be trying to get fluid that's outside of the blood vessel in the interstitial tissue fluid space. That's going to be trying to push and get into the blood vessel. But like I said, it's pretty much zero. And when it's not zero, there's some sort of pathology going on, right? Some sort of pathology. Okay, so those are the two types of hydrostatic pressure. The other type of pressure is the colloid osmotic or oncotic pressure. And so we kind of describe this as the pull. Right? It's almost like a push-pull relationship. And of course, we're going to be dealing with water because that makes up the majority of the fluid that's in the blood and usually outside of the blood vessels there. And so what is going to generate that pull? It's going to be the proteins, primarily the proteins. So that's when you see this word here, colloid, you should be thinking protein. When you see osmotic, you should be thinking water. Okay, so that's what we're talking about here. All right, so the colloid osmotic pressure is going to be that force generated by the proteins to attract or pull water towards wherever those proteins are. So if there's a lot of proteins in the blood vessel, then it's going to be that force pulling water into the blood vessel. If a lot of those proteins are outside of the blood vessel in the tissue, in the interstitial fluid, then it's going to draw water, all right, into the interstitial fluid, all right, from the blood vessel. All right, we'll talk about that. So the first one here is the blood colloid osmotic pressure, right? COP subscript B. Right, so again, this is that pulling attractive force, right, that the proteins that reside in your blood are going to be exerting on water. So it's essentially going to try to pull water into the blood vessel, or you could say it's holding water in the blood vessel so it's not escaping or leaking out. And we talked way back in chapter 18 that this class of proteins, the albumins, are the primary protein class that exert this blood colloid osmotic pressure. And what organ is responsible for making those proteins? Take a guess. Liver. Yeah. Okay. Yeah, the liver. That's why you'll see with folks that have liver damage, i.e. chronic alcoholics, right? One of their symptoms you'll see that they'll have edema, tissue edema, pitting edema. And that's because they're no longer producing the albumins like they could. And so water's essentially not being pulled back into the vessel. So it's able to leave the vessel a little bit easier and move into the tissues. I would get into this in more detail, but we're not going to be covering that chapter this semester, so I'm sad about that. Okay, so right, when we talk, you'll hear me go back and forth between colloid osmotic pressure and this term here, the oncotic pressure. Just kind of keep that in mind. It means the same thing there, all right? So our blood colloid osmotic pressure is going to favor resorption. Resorption, that means we're going to be pulling things into the blood vessel there. All right, so interstitial fluid colloid osmotic pressure. It's a handful of words there. COP subscript IF, right? This is going to be, right, what influences the movement of water in the interstitial fluid. Okay, and so again, we're going to be dealing with another situation in which we have a very low pressure out in the interstitial fluid. Because technically, you should not have any proteins in the interstitial fluid. I shouldn't say any, all right, but you should definitely have a significantly less amount of proteins in the interstitial fluid. Why? Because it all should be in the blood. And so what we'll see then, if there's hardly any proteins out there, they can't exert that osmotic pull and they won't be drawing water, all right, from the blood vessel into that tissue there. Right, so in a normal individual, it should be between 0 to 5 millimeters of mercury there. Right. So again, very few proteins. And the thing is, and I want you to understand this, and I know I've mentioned this before, the composition of your blood plasma and the composition of the interstitial fluid are pretty much identical, except you have much more protein in your blood and hardly any or very few in the interstitial fluid. Okay, so the composition of blood plasma and interstitial fluid are virtually the same, except there's more proteins, or you're going to have a lot of or proteins in the plasma and not so much in the interstitial fluid there. Okay, so now this leads us to this nice fancy equation that we're all going to be responsible for, all right, on our next test. This is the net filtration pressure. All right, so there's the equation there. So net filtration pressure is equal to the difference of both of your hydrostatic pressures, right? And then it's also going to be the difference of both your colloid osmotic pressures. And then we're just going to subtract your colloid osmotic pressure from your overall hydrostatic pressure. That's what the net filtration pressure is. So like the definition says up there, NFP is the difference between net hydrostatic pressure and net colloid osmotic pressure. So that's what we're going to see here. So we're going to take the hydrostatic pressure both inside the blood vessel, outside the blood vessel, take the difference of that. And then we're going to take the colloid osmotic pressure both inside the blood vessel and outside the blood vessel. We'll take the difference of that. And then we'll take the difference of those two values from one another. And if it's a positive value, then we favor filtration. And that's going to be, or it should be, at the arterial end. If it's a negative value, then it's going to favor resorption. And that should be seen at the venous end there. So as we move from the arterial end of the capillary to the venous end of the capillary, you should see the net filtration pressure start to decrease. It will change. So it'll be high at the arterial end and very low at the venous end. But by design, because we want to see that filtration occurring there. We want to see filtration occurring at the arterial end to get stuff out of the blood, and we want to see resorption occur at the venous end to pull the stuff, the wastes, and all the other solutes that the cells have produced during their metabolic activities, right, and dump it into your blood and get rid of it. All right, because that's what your bloodstream is going to be. That's going to be, I already went over this in the last class, so I won't go over this per se here. But again, here you can see, let me get rid of this. Here you can see, right, as we move from the left side of our screen to the right side of our screen, you'll see here's the filtration, the hydrostatic pressure inside the blood, right, by the arterial end. But by the time we get all the way over here to the venous end, it's dropped significantly. But again, it's always going to be pushing outwards. And then you'll notice here the interstitial, all right, fluid osmotic pressure, right, relatively stays the same because those values should not change. The only way they should change, right, and especially when we're dealing here with the colloid osmotic pressure in the interstitial fluid, is if proteins are leaking into the interstitial fluid. And in that way, it would increase. Okay. All right, so that's kind of a little bit of a review of the high bulk flow. And so you should expect to see some of that revisited here when you take your tests. Right, so just kind of go over that, listen to my video if you're not sure, read the chapter section on that if you're still having problems. All right, so quick blurb about the lymphatic system, because since we are talking about fluid in the interstitial tissue space, I do need to mention that the lymphatic system, and we'll talk more about it more so in lab, but I want you to think of the lymphatic system as kind of like the sewer system, or like what we just saw not too long ago with the storm that came through. All right, it's the storm drain. So you'll get an increased amount of fluid that has come to an area. In this case, you know, when the hurricane came through, we saw a lot more water. And so where does that water go? It goes into the storm drains. All right. And so here, our lymphatic system is going to act like the storm drain. It's going to drain off the excessive fluid that was not resorbed by the capillaries. And that's usually around 15% or so. Right. But in a different situation, like if you sprained your ankle, you'll get an accumulation of fluid in the interstitial fluid space. And that's because sometimes there's proteins that are stuck in there. And so you'll see more than 15%. But the lymphatic system plays a big role. It's eventually going to drain that fluid off of those tissues. And then the nice thing about the lymphatic system is it winds up dumping all of the lymph back into your bloodstream. And then it gets processed, and we'll learn about that as the semester progresses, where that lymph goes, that lymph fluid goes. Okay, so now the next concept, local blood flow. Right, local blood flow. So you have capillaries everywhere, feet, right, nose, ears, stomach, wherever. It's all over, every tissue. Not all of those capillary beds are going to have blood in them. Remember, talked about this as concepts. We're going to keep revisiting it. Supply and demand. Supply and demand. Your body will supply areas that need or are demanding that fluid or blood flow to that area. So obviously, for example, exercising muscles. If you're going and working out in the yard, removing storm debris, or if you're going for a run, right, your skeletal muscles demand more blood flow to supply it with nutrients, oxygen, with glucose, so those cells can metabolize, so they can make energy, all right, so they can perform whatever work that needs to be done, okay? So the other tissues might not, like when you're sleeping tonight, okay, your skeletal muscles don't need a lot of blood flow, okay? And so we'll see that the blood is going somewhere else, and so those capillaries will not have, all right, the normal amount of blood flow as to what they would have when you're exercising. So keep that in mind. So not all the capillaries are going to be filled simultaneously. And so when we talk about our local blood flow here, right, we're going to measure that flow, right, with a couple of different values here. We're going to measure it with a volume value and then a time value, right? So milliliters per minute. So when we talk about this, that should look familiar to you because I talked about the concept of perfusion. And perfusion is essentially, right, blood flowing to a tissue, all right, a specific square, I don't want to say footage. It's going to be a specific amount of tissue that is going to be perfused or have blood flowing to that tissue, right, over a specific time. Certain volume of blood is going to go to a specific area of tissue over a specific period of time, which is going to be 1 minute. We always like to do 1 minute. So that's pretty much what we're going to be looking at here. So we're going to talk about the 4 things that influence this local blood flow. So what is causing blood to go to this area and not to another area? So as you can see up there, there's 4 things, all right? Degree of vascularity, right? Myogenic response, which is, think of myo being muscle, so myogenic response, so there's going to be some sort of muscle tissue influencing this, right? Other regulatory factors, and then what we call total blood flow. So we're going to spend a lot of time on total blood flow. All right, so let's move through. Let's talk about the degree of vascularization. All right, so hopefully in 210 you learned that certain tissues are more vascular than others. Like, for example, mature cartilage, avascular, no blood flow to mature cartilage. But bones are highly vascularized. So different tissues, different organs, right, are going to have varying degrees of vascularity there. So obviously, if certain tissues are more metabolically active, they should have more blood vessels. Yeah, they should have a higher vascularity. So if you look at the examples up there, every single one of those should make sense to you. Okay, brain, skeletal muscle, heart, and liver. Those are all relatively highly metabolically active tissues. Some it varies depending on whatever activity is occurring. Right, so we know that the skeletal muscle is going to vary, right, in its metabolic demands depending on what it's doing. Like when you're sleeping tonight, very low demand. When you're walking down the stairs, a little bit of a higher demand, right? Same thing with the heart, right? So keep that in mind. So now when you look down here at these tissues here with low vascularity or that are avascular, you should have learned that tendons and ligaments, those are made up of dense, regular connective tissue. Dense, regular connective tissue has very little space for blood vessels, so it has a lower vascularity, takes longer to heal, for example, right? So certain other tissues, like I mentioned before, mature cartilage is also avascular. So you'll have low to avascularity depending on what the tissue is. So when we talk about the degree of vascularization, that's what we're talking about, okay? And mainly it varies on the function of whatever that tissue is. All right, so this term here, be familiar with it, angiogenesis. Angio has to do with blood vessels. So angiogenesis is the formation of new blood vessels. So when does this occur? Right, well, in certain situations, like when you look at your professional athletes, and you can see it on some people. Some of these professional athletes, if you see them walking by, you'll see all these veins in their muscles, all right? And so because they've increased their muscle tissue mass, what do we call that? There's hypertrophy, and then there's also hyperplasia. The big thing is hypertrophy is an increase in the cell size. Hyperplasia is the increase in cell number. So you can get an increase in tissue mass through those two ways. Atrophy, though, is both decrease in cell size and a decrease in cell number, right? So we don't have a different term for that. All right, so when athletes are training, they're going to get an increase in their skeletal muscle tissue. And so we need to supply that demand, right, that those skeletal muscles are going to require from that athlete. So we'll see an increase in vascularity there. Same thing with adipose tissue, okay? Folks that have gained an increase in adipose tissue, and I've already figured this out, if I were to ever go on Survivor, I'm going to pack on 50 pounds. Because then I don't have to worry about trying to win those challenges to eat. I'm just going to live off my adipose. And so what you're going to see is what are the purposes of adipose tissue? Cushioning, insulation, but most importantly, energy storage. Energy storage. We've got to get that energy out to the other tissues that need it. And so we're going to grow blood vessels in that new adipose tissue so we can release those lipids and whatnot, and those triglycerides for energy metabolism there, all right? Coronary vessels. This one here we talked a little bit about when you saw some blockages occurring. And so you might hear about somebody that had a coronary bypass surgery, and you'll see that the blood vessels that they had to replace were significantly blocked. You'll hear numbers like 80%, 90%, and you're like, how did they live? Well, a lot of the times, right, those coronary vessels would create anastomoses. You'll get these new blood vessels that will connect to one another. Now, it's not enough to meet all the demands of the heart, but it surely slows down, right, the symptomology of that individual there. So you'll see these types of situations in which we get an increase in these new blood vessels, and the whole purpose of that is to increase the tissue perfusion. We got to get more blood out to those tissues because they need it for whatever their metabolic functions are going to be. Okay, so that's angiogenesis. So, like the saying goes, what goes up must come down. So you have angiogenesis, the formation, well, then we'll have regression. And so with regression, it's just the opposite. So this is when we go back to our normal state. So say that elite athlete is no longer competing anymore. They've retired from their sport, or they got injured and they had to stop, right? So when they stop their training, then the demand's not there anymore. So the skeletal mass starts to decrease. And so therefore, if the skeletal mass starts to let me be specific. If the skeletal muscle mass decreases, then that demand isn't there. So we don't need the blood vessels to supply that tissue. So we'll start to see a regression in the blood vessel formation. Same thing, you'll see the individuals that lose a lot of weight, all right, decrease the adipose tissue, all right, content, then there's no need to have all those blood vessels there. So we'll start to get rid of the blood vessels there. And that's a great concept there, because we'll talk about the resistance and whatnot later on in this chapter about blood pressure, because we're going to talk about some really interesting concepts when we get to blood pressure. And remember, I said blood pressure, one of the equations, it's not one of the required equations that you have to know, but blood pressure is equal to heart rate times total peripheral resistance. Well, what we're going to see is the larger your how do I want to say this? The larger the blood vessel volume that you have, the more blood vessels that you have, you will increase your total peripheral resistance. I'll get to that concept here. I don't want to blow your minds too much. All right, so here, our first clinical view here, tumor angiogenesis. I had a colleague that had a very rare form of eye cancer. She had an ocular melanoma, and her type of melanoma has a high chance of spread. And when it has a high chance of spread, there's a high chance, obviously, of metastasis and then death. Well, anyways, when she undergone radiation treatment and has the tumor no longer has a blood supply to it. And so tumors are really clever, because as they grow in cell size, they're eventually going to want to gain more resources. And so they can essentially get resources through diffusion, essentially, but that's not enough to sustain uncontrolled growth. And that's what tumors like to do, uncontrolled abnormal cell growth. And so what we'll see is some tumors get clever, right? They can, one, evade the immune system, and so they stay alive because the immune system is not going after them. And then two, they're like, well, I've run out of these resources here. I've got to get my own resources, so I'm going to try to get some blood vessels to grow to me, so the body is going to supply me with blood so I can keep doing what I'm doing. So some of these tumors here, they'll produce what they call growth factors. So we talked a little bit about growth factors, remember the colony stimulating factors that we talked about with hemopoiesis? Well, what we'll see is you have these growth factors that tissues will produce that will kind of stimulate the formation of new blood vessels. The tumors start to learn how to do this, and they start producing these growth factors. And so they'll start to cause blood vessels to grow towards the tumor. So they're looking into, all right, ways to decrease growth factor production that will starve the tumor out. And so we're kind of seeing that with research and whatnot, but they haven't really quite gotten it where they'd like to be. Okay, so let's talk about the next type of local blood flow, and this is the myogenic response. So again, I said myogenic, you should be thinking muscles, but specifically, I want you thinking smooth muscles. Why? Because your smooth muscles are in your blood vessel walls, right? Because the smooth muscles control the size of the lumen, right? So when the smooth muscle cells contract, they decrease the lumen size, they increase resistance and decrease blood flow, and vice versa. When the smooth muscle cells relax, right, they'll increase the size of the lumen, and then that will decrease resistance, which will increase blood flow, right? And so we'll talk about this. But what I want you to understand about the myogenic response is the myogenic response is trying to keep things constant, right? It's like a typical male. We don't like change, all right? And so the myogenic response is like a male response. We want to keep things constant, so not changing. So that's what we're going to see here. And I'm going to give you some examples to kind of help explain this. So you want to keep that thought in your head that when we're dealing with the myogenic response, we're trying to keep our blood flow at a constant rate, at a constant amount, right? So the first example that you see up there is what happens if our blood pressure increases. So as a result of that, right, we're going to see more blood going into the arterial. And so when more blood is going into the arterial, it is going to put more of a stretch on the arterial wall. So you have baroreceptors. Those are the stretch receptors that we find in our blood vessels. And so those baroreceptors, all right, will then transmit that information of the stretch occurring there. And so what we're going to see is, again, we want to keep blood flow constant. We just got a surge in blood flow. So we want to now decrease it a little bit. How do we do that? The smooth muscles will vasoconstrict. That vasoconstriction increases resistance for a period of time, decreases blood flow back to the original levels. That's what we're trying to do. Right? We want to keep everything constant. So as systemic blood pressure goes up, you're going to see, all right, more blood coming into the arterial, which is going to put more stretch on the wall initially. All right, and so the response is going to be to contract the blood vessel wall, causing an increase in resistance, which is going to cause a decrease in the blood flow, right? Until we get it back to the original levels. So the same thing when we go the opposite direction, when we see a decrease in our systemic blood pressure. So now we're going to see a decrease in the amount of blood flow. So therefore, we'll have less stretch on the blood vessel walls there. And so in response to that, we're going to get vasodilation. And vasodilation, right, causes the blood vessel wall to relax, and that will increase blood flow because we decreased the resistance momentarily. So again, going back to those constant, consistent levels there. So it's almost kind of like a negative feedback response. But again, with the myogenic response, I can't stress it enough. A couple things you want to know. It involves the smooth muscles of the blood vessel walls, and its goal is to maintain consistency, right? We want to keep that local blood flow to those tissues at a consistent rate and amount, I should say. Okay, so how, all right, when we talk about our local blood flow here, we're going to talk about some local short-term regulation now. And how, when we talk about locally regulated, I'm not talking about involving the central nervous system, okay, the vasomotor center. I'm not talking about that. This is going to be on a tissue level, what's happening on the tissue. And again, you can see these three important words: based on need, supply and demand, supply and demand. Okay, so what is that need? Well, obviously, one of those needs that we talk about all the time is that metabolic activity, right? So what are the changes that can occur with metabolic activity or if the tissue becomes damaged? So we have these chemicals, right? We call them vasoactive chemicals because they are going to influence the blood vessels here. And so how are they going to change that blood flow there? So you have two types, the vasodilators and the vasoconstrictors here. And so these vasodilators, obviously, right, when the tissue releases the vasodilators, right, or if those vasodilators are present in that tissue, that's going to cause the smooth muscles to relax, and it's going to cause the lumen to become larger, all right? And so we'll see, especially, remember those precapillary sphincters? I don't know if I have a picture of it here. Yeah, kind of. Well, it would be right here, the precapillary sphincter, one of these things. Smooth muscle cell. All right, so those precapillary sphincters, where'd I go? Right, it's going to cause those precapillary sphincters to relax, therefore providing less resistance, therefore allowing, all right, more blood flow to that area there. All right, the vasoconstrictors, just the opposite. They're going to cause constriction here. So they'll cause vasoconstriction in the arterioles. And so what we'll see now is contraction in those precapillary sphincters, which is going to decrease blood flow. So first I want you to understand the two classes, vasodilators and vasoconstrictors. And we're going to talk about what some of these are here. All right, the next concept, all right, for our local blood flow is this autoregulation. As you can see, there's a lot of stuff that goes on in your body that you don't even realize. This is all happening as you're sitting here. Not so much unless you're exercising and whatnot, but as you're sitting here, some of this is occurring here. All right, so autoregulation. Here we go. Process by which tissue controls local blood flow. So autoregulation is almost like the autocrine response for your hormones. Remember, we talked about the different ways that hormones can affect, right? There's paracrine, autocrine, right? And then we have, I'm forgetting the third one. That's awful. My brain's working at 100, I know. So autocrine is when the cell releases the hormone that acts on itself. Did you say Merrick? No, is it paracrine? I can't remember. This is awful. It'll come back to me. I'll be sitting in my car driving home. All right. So let's look at this example here. All right. Tissue activity increases. All right. So when tissue activity increases, we're going to see what's going to happen. That demand increases. And so what we'll see is those tissues, if they're not getting perfused enough, if blood flow is not flowing like it should for whatever reason, right, we are going to have these vasodilator substances being released. We just talked about our vasoactive and the vasoconstrictive there. So the vasodilators, these substances will act, all right, as vasodilators. So for example, right, you'll see a decrease in oxygen presence, a decrease in, let's say, glucose or amino acids. When those all go into decline, they will act as a vasodilator on those capillaries. And so they will increase tissue perfusion because they'll increase blood flow. Conversely here, we'll see if we get an increase in carbon dioxide, lactic acid. If you all know what lactic acid is, it is when you break down a glucose molecule into what we call pyruvate. If there's no oxygen present, then your body converts pyruvate into lactate or lactic acid. Everyone knows lactic acid more commonly because it makes their muscles sore. And usually it's because it accumulates in the muscle tissue and it causes that soreness sensation there. All right, but if you look here, carbon dioxide, lactic acid, hydrogen ions, right, and also potassium, all of those, if those increase, will also act as a vasodilator. And so they'll increase tissue perfusion through blood flow. And so this is all based on that negative feedback system there. And so as perfusion, right, increases the area, and now we've adequately met the demand of that tissue, then though the levels of oxygen will increase because it's coming in on the blood, right, and also the nutrients. So these levels will increase, and then carbon dioxide, lactic acid, and the rest are going to decrease because they're being carried away. And so then we go back to the original set point with our negative feedback here. So that set point, we get there by vasoconstriction here. So you can see this in this reactive hyperemia. Okay, reactive hyperemia, well, you don't see it down here as much, but you see it up north all the time. Somebody would be outside where it's freaking cold, and they step into a nice house place that's like 75, 80 degrees inside, and you'll see their face get red and flushed. And that's because you get that change in temperature causes, all right, that vasodilation in the blood vessels and the superficial blood vessels in the skin. And so that causes an increased blood flow. And we all know that one of the things that gives color to your skin is the amount of blood flow going through there. And so usually folks that have increased blood flow in the skin will appear flushed. And so that's what we will see here. Do it to yourself. You can just press down on your fingernail, hold it for 10 seconds and then release. And you can see initially when you raise your finger off of your nail, it's white and then it quickly turns red. And that's a great way to actually assess somebody's circulatory system by just pressing down on that. The color of the nail bed should instantaneously return back to that red or pink. If it takes longer than 10 seconds, you need to have that person checked out, right? Because they're having compromised blood flow to that tissue there. Okay. So one of the things that we're going to see, I mentioned two things here when I was talking about, locally regulated. One was the metabolic activity, what we just talked about. And then the other one was what happens when the tissue is damaged. And this gets us into inflammation. We'll talk more about inflammation when we get into the immune system, right? But we all are somewhat familiar with inflammation here, right? We're going to see the the tissue, and it's going to be, it varies depending on how damaged that tissue is, but inflammation is going to bring in our leukocytes and our platelets, right? In addition to the damaged tissue here, all of these structures will release some of our vasoactive chemicals here. And the two that we're familiar with histamine. We saw that the basophils and the mast cells release histamine and heparin. And so histamine is essentially going to make your blood vessel leaky, right? And then bradykinin is going to also contribute to, and histamine does the same thing, but they cause dilation here in the arterioles there. And so when you get increase in dilation, you're going to get an increase in the blood flow. And so, of course, we want to increase our blood flow to that damaged site to bring in, all right, things to repair whatever was damaged, but also, all right, whatever's causing that inflammation, it could be, if it's a pathogen, like a virus or bacteria, we'll bring in our white blood cells to help kind of fight whatever that is. But don't just blame it on trauma, but allergy, for example, one of the reasons why we take antihistamines like Benadryl, all right, is to help decrease the histamine production there. All right. Huge thing that we don't really talk about too much in this course, but nitric oxide is an impressive vasodilator, and nitric oxide can be made by certain cells with certain chemicals to cause that vasodilation. I know for a while, a lot of bodybuilders used to take I shouldn't say nitric oxide, but the components that would generate nitric oxide in their body. So they take a certain kind of shake and then drink it down. And then within a half an hour, once it metabolized, it would help the cells and the tissues produce a lot of nitric oxide. Why? Increases vasodilation, increases blood flow. So they get a better, what they call a pump or workout there. All right, so we said that we see tissue being vasoactive chemicals for dilation. What about constrictors? So these two examples, prostaglandins and thromboxanes, should look familiar to you. You all remember where you saw those from? Nailed it. Yep, locally acting hormones, eicosanoids. They all come from arachidonic acid. So prostaglandins, thromboxanes, leukotrienes, right? Those are eicosanoids there. So they're on hand on the cellular level here. And so these substances here, right, when tissue becomes damaged, they can release, right, our prostaglandins and thromboxanes, and there's different subclasses for those. And so they'll act as vasoconstrictors. Why? Prevent blood loss, right? And again, it's dependent on the extent of the damage. So some of you are thinking, well, what the heck? It seems like you have conflicting situations going on here. We got vasoconstrictors and vasodilators, and they're all working, at the same time, or, what's going on? Yeah, that's true, right? You're going to have, right? Obviously, the vasoconstrictors are going to be in high concentration to decrease, right, blood loss, depending on if there's blood vessel damage, because there doesn't necessarily always have to be significant blood vessel damage, but that's the decrease. And we learned about hemostasis there, about the stopping of our blood. And so that's what we would see in this situation, right? But at the same time, we also need to have our vasoactive chemicals here to help promote dilation, so we can get, all right, components from our bloodstream, like white blood cells and platelets and a lot of other proteins there, to help, right, mitigate any tissue damage that's occurred and also decrease inflammation. This is why I tell folks all the time that inflammation isn't necessarily a bad thing, like a fever. And we'll talk about that when we get to the immune chapter, that a fever isn't bad unless it's like 108, 106, then it's bad. But a 99 or what we call a low-grade fever is not necessarily a bad thing. Now, if it's like that for several weeks or months, then there's an issue going on, right? Inflammation is the same thing too. A little bit of inflammation, acute inflammation, is good initially to the response of whatever the damaged tissue was. But if you have chronic inflammation, like months and months out, that's bad. All right. And we'll touch on all that when we get to later chapters here. All right. The last concept that I want to talk about in regards to our blood flow here is what we call total blood flow. Now we're going to talk about, all right, the amount of blood that is going to be moving through our vasculature. And so if you look at that next bullet point up there, equal to the cardiac output. So pretty much, and we discussed this, I believe, in chapter 19, that pretty much all your blood in your body gets pumped within a minute, right? Your heart's essentially going to put out your total blood volume, right? Because cardiac output is going, does anyone remember the equation for cardiac output? CO equals what? Yep. Heart rate versus stroke volume. Heart rate is based on beats per minute. So again, this is going to be another, right, unit of time that we're going to use that is going to be a minute. So, and when we equate it and factored it out in our previous class, we said it's about 5.25 liters per minute, which is usually about the blood volume for an individual, for both male and female. Usually males have a little bit more. So when we're talking about our total blood flow, right, this is what we want to see. We want to see our total blood flow equal to our cardiac output. Now, obviously, this is going to change depending on what your tissues are doing. All right. If you're like we're all right here sitting, it should roughly be about 5 liters per minute. Now, if we start to exercise, that will change significantly because, and just look at that equation, cardiac output equals heart rate. So what's going to happen when you exercise? Your heart rate increases. So that's one way, all right? Your stroke volume will also increase too, but your heart rate will increase, so therefore your cardiac output should increase, which means then your total blood flow should also increase because if your heart's putting out more blood, you're going to get more blood flowing through your vasculature here, right? And so what we're going to see, again, keep in mind, it's all dependent on the demand of our tissues here. Right, so when we talk about total blood flow, like that last point down there, this is important. Regulation of total blood flow depends on both the heart and the vessels. So this is kind of getting us close to blood pressure. Right, so if your heart increases, then your heart increases. If your heart rate increases, your total blood flow should increase. If your stroke volume increases, your total blood flow should increase. Right, so we're going to see that there's going to be some similarities there. Right. So this brings us into, all right, I say this is the good stuff, right? Because we're going to start getting into blood pressure and what is occurring, right? What the purpose of blood pressure is and blood flow and whatnot. So this graph here, I'm going to be going back to this, all right, I shouldn't say graph, this figure quite frequently. So what this is showing us here, right, is on the bottom here, on the x-axis, you can see, right, there's different blood vessels here, starting with the aorta. So this is closest to the heart. And then we're moving away from the heart. We go to the arteries, we go to the arterioles, we go to the capillaries, and then we're going to start coming back to the heart. We go to the venules, the veins, the vena cava, superior and inferior vena cava, into the heart, and then over again. You got to kind of see what on here on the x-axis, all right? So these are the different blood vessels. We start near the heart, then we go away from the heart, then we come back to the heart. Then this is on the y-axis, that's our blood pressure there. So you'll see, all right, initially, when we're close to the heart, when we're in the arterial system, when we're dealing with the aorta, the arteries, and even the arterioles, you'll notice that the pressure, the blood pressure changes. And we know this from our own blood pressure. What I mean by this, when you go in and get your blood pressure checked, what's the average blood pressure volume? 120 over 80. Yeah, okay. So 120, the top number is the systolic. We know what systole is. That's when the heart contracts. The bottom number, the lower number, is the diastolic. That's when the heart is relaxing. So you'll see here on our figure, our systolic pressure is around 120 here during systole when the heart's contracting. But you'll notice it drops down, right, to 80 when the heart's relaxed. What's one of the things that you're noticing? So this is the difference in between here. You'll notice as we're in the aorta, there's a pretty big difference, right? But what are you noticing as we move further away from the heart? What are you noticing about these two lines here? Right, they're getting closer and closer together. The difference between the pressures becomes less and less. And by the time we get to the capillaries, there's no difference in pressure. And then when we're in the veins, again, the same thing. But if you notice, check across the board. Here to here, what are we seeing overall? What's happening to your blood pressure? It's decreasing. Yeah, okay, it's decreasing. So keep that in mind. I just want to point out some of these concepts here. One is as we move through, all right, close to the heart, and then as we move away from the heart, right, through into the venous system, our blood pressure just gets less and less and less and less. In the arterial system here, in the aorta, the arteries, and the arterioles, right, the difference between the systolic pressure and the diastolic pressure becomes less and less and less. So again, these concepts will come in later on. I just want to draw that to your attention there, because you're seeing there's this guy right here called the MAP, the mean arterial pressure. And so this right here, we're going to calculate that out. I'll talk to you about that in a second. That's this line here, this red line here. All right, so let's talk about the arterial blood pressure. And that first thing I was saying is the MAP, the mean arterial pressure. So the mean arterial pressure, the definition is the average arterial blood pressure across the entire cardiac cycle. What's the cardiac cycle? Loved up, one heartbeat, right? Okay, so that's the cardiac cycle. Everything that we learned in chapter 19 about blood moving, right, into the atria and then going into the ventricles and then getting pumped out of the heart. That's what we're talking about here. So when we talk about the mean arterial pressure, we're going to just talk about the pressure in your arterial system there, right, as the heart beats. It's essentially what it says. Okay. So I'm going to skip around on the slide here a little bit. MAP, the mean arterial pressure, that's the definition. Okay, so what you're going to do is you're going to take the diastolic pressure. So we saw on this slide here, our diastolic pressure was what? 80. Okay, good. And then what you're going to do is you're going to take the pulse pressure. What the heck is that? I'm going to tell you. The pulse pressure is the difference between the systolic pressure and the diastolic pressure. So you're going to take, what's the difference of 120 minus 80? 40. You're going to take a third of that. So divide it by 3. And that should give you what, 13.3 or something like that, if anyone's doing the math. Okay. Something like that, right? So that's what we're going to see here, right? So in case you want to know, right? So diastolic pressure is going to be 80. And then we're going to take 1/3 of the pulse pressure, which is the difference between, if the blood pressure is 120/80, so that difference is 40. You're going to just divide 40 by 1/3. Or you should multiply by 1/3, but actually you're going to divide it by 3. So if you take, all right, 40 divided by 3, it's roughly going to be 13.33333, I believe. And so we're just going to add those two values together. So 13.333, just drop the decimal point. So 13 plus 80 is 93. All right, and so this is basically going to tell you, all right, perfusion. If it's above the value of 60, we're happy. If it's below the value of 60, we're not happy, right? Your tissues aren't happy. All right, so that's going to be our mean arterial pressure. I want you to be able to calculate that out. Okay, so diastolic pressure plus 1/3 of the pulse pressure. And just to remind you, the pulse pressure is the difference between, all right, your systolic and your diastolic pressures. All right, so that's the mean arterial pressure. So now, going back to this picture here, as we get further away from the heart to the capillaries, we see that our blood pressure starts to get less and less, and it starts to get closer to the 0 value. But in the capillaries, there's still some pressure going on. And you can see from the arterial end of the capillary to the venous end of the capillary, you'll notice, all right, it falls off a decent amount. That's the hydrostatic pressure. So that's why we favor filtration here, but favor resorption over here, because part of the reason is we're seeing a decrease in the hydrostatic pressure. What influences the hydrostatic pressure is your blood pressure there. So that's what we're going to kind of see here. All right. So the important thing is, by the time blood is in the capillaries, it's not going to be fluctuating. Because remember, when your heart beats, you get a little bit of a shove on your blood. And then when the heart relaxes, right, it just kind of lingers or kind of continues on with the flow. But every time the heart beats, it gets a push. It gets a push. So by the time you get down into the capillaries, it's not fluctuating. It's nice and constant. It's a nice, easy, rhythmic flow there, right? Here's the problem. When we're in the capillaries, we need that pressure to be high enough so you can have filtration. So we can move stuff from the blood vessels, from the capillaries into the tissue, right? But it needs to be low enough as to not hurt the blood vessels. So that's why people with high blood pressure were so concerned about them. Because high blood pressure over a period of time can cause damage to the blood vessels, especially to the blood vessel walls. One of the things that we're concerned, what's one of the things that you're concerned about somebody with really high blood pressure having? Think your brain. Well, headache's a symptom of it, but stroke, yeah, right? And stroke could be from an aneurysm, burst of blood vessel, that's a problem, right? So we don't want our pressure to be too high. So when we're at the arterial end of our capillary, we'll see about a 40 millimeter of mercury pressure. By the time we get out or towards the end of the capillary near the venous end, we'll see 20 millimeters of mercury. That's what we're seeing here. It's about 40 over here at the arterial end. And then by the time we're at the other end of the capillary by the venous end, it falls to 20. But notice it's one single line. These two lines should indicate to you that it's a pulsatile type of movement of the blood. Okay, so every time the heart beats, it pushes the blood. Then when the heart relaxes, we're still going to see some blood flow, but it won't be, as much as when the heart was contracting. All right, so it's pulsatile here. That's why we take the pulse on arteries and not veins. Veins don't have a pulse, right? Okay, you can test it out. Push on one of your veins and see if you can feel the pulse. You won't be able to because, again, it's all going to be one single line there. Okay. All right, so let's talk about now venous blood. Okay, I told you I was going to keep going back and forth. Now we're going to talk about this part here. Notice there's no pulsatile movement, but as we go from the capillaries back to the vena cava, your blood pressure is continuing to decrease in the venous system. So some of you might be thinking, well, how the heck does blood get back to the heart? We're going to talk about that, right? Because if your blood pressure keeps falling, right, how is it going to keep moving through your vasculature? We're going to get to that, right? We're going to get to that. All right. So this is where we talk about venous return. Where are you? All right. So venous return, if you remember from chapter 19, venous return should equal, right, your stroke volume, okay? Or I shouldn't say, or your stroke volume. The amount of blood returning to your heart should equal the amount of blood leaving your heart. We agreed upon that, right? We talked about the balance that you should have. Both your right and left ventricles should be pumping out the same amount, all right, of blood each beat. So your stroke volume should be equal on the right and left ventricle, okay? And the amount of blood should be equal returning to the heart, right? So when we talk about the amount of blood coming back to the heart, it's dependent on three things. One, the pressure gradient. Two, this thing called the skeletal muscle pump. And three, the respiratory pump. And we'll go through all these. We're going to go, I'll talk about these here. All right, a couple things I don't want you to forget, and I already said this before, and I'm going to repeat myself. One, all right, our venous pressure is non-pulsatile. Remember, we're not getting a pulse in our veins. You'll get it in the arteries, but not the veins. And it's very low. See, it's decreasing as we move towards the heart. It keeps getting less and less and less and less. So I want you to understand those concepts because we're going to talk about them here in a little bit more detail. So you'll see, well, remember pressure gradients, high pressure, low pressure area. This would be a high pressure area considered to this spot here. That's a low pressure area, right? So the pressure is higher on the capillary, on the venous end of the capillary, and it's lower at the vena cava. So some people have a hard time swallowing that because they're thinking, well, the capillaries are so small. And the vena cava and the veins are much larger than that. How is the pressure falling? We'll discuss. All right, so let's start off here, right, with our first, mechanism here called the skeletal muscle pump. So the skeletal muscle pump is important, and you hear it when you're flying on an airplane, right? Don't sit, especially if you're on an overseas flight where it could be as long as 17 hours in some cases. They tell you don't sit the whole time, get up and move around. Why? Because when you're sitting there, blood pools in your legs. It's not going anywhere. We just saw on that graph how the pressure keeps going less and less and less, right? Even as we get closer to the heart, right? Or the vessels get closer to the heart. So one of the ways that we've managed to get blood to move from your upper extremities and your lower extremities, primarily the lower extremities, is the skeletal muscle pump. The skeletal muscle pump, remember, veins have this very important structure. Valves, okay? And we know from the heart that valves prevent backflow. And so the veins are going to prevent backflow, or excuse me, the valves in your veins are going to prevent backflow. So how does this work with the skeletal muscle pump? Well, the skeletal muscles start to contract. They push on the blood vessel, and blood, all right, is going to get squeezed. Well, I'll show you the picture here. As the muscle contracts, blood gets squeezed in either direction. It can either go upwards here to the heart, or it can go down, all right, down to the lower portion of the lower extremities, for example. Well, guess what? We got this vein here that prevents any backflow. So it can't go down. It can only go up because the valves only open in one direction. Then when the muscles relax, right, then it decreases that pressure on the vessel wall. So this is what happens, and y'all know when you walk around, right, your muscles contract and relax and contract and relax. And so it almost acts like a pumping mechanism. So when you're walking around on the airplane, those muscles in your legs are pumping or contracting and relaxing, and they're pushing that blood in your lower extremities up towards your heart there. And so same thing happens when you're exercising or any type of thing. So this is the skeletal muscle pump. The respiratory pump, similar type of mechanism here because it's going to involve pressure gradients again. And so this is how we're going to get blood to move back towards your chest here. And so this has to do with when you're breathing in and when you're breathing out. So when you're breathing in, and we'll talk more about air pressure gradients when we get to the respiratory chapter, but when you take a breath in, this is what happens. Your diaphragm flattens out. The muscle contracts. Normally your diaphragm's like a dome. And so when you take a breath in, that muscle contracts and it flattens out. And so what that does to your chest cavity is that it increases the diameter of your chest cavity from front to back, from side to side, and from top to bottom. Again, we'll learn all this, so don't worry about memorizing that. But here's what happens. You're going to see as the diaphragm contracts, the pressure in your abdomen increases, and then the pressure in your thoracic cavity decreases. That's how breathing essentially works, right? When you take a breath in, your chest cavity expands, and the pressure inside of your chest, inside your lungs, becomes less than the air pressure in this room. So what will happen is air moves down a pressure gradient. It moves from an area of high air pressure to an area of low pressure. It enters in your mouth. We'll talk about that. But what we'll see here, since you have an increase abdominal cavity pressure and a decreased thoracic cavity pressure, where does blood want to go? From the area of high pressure to the area of low pressure. So the blood in your abdominal veins, okay, inferior vena cava, renal veins, all of those popular veins there in the abdomen, blood from those veins will move towards the thoracic cavity. Then when you exhale, okay, the opposite happens, all right? The diaphragm relaxes, but now the thoracic pressure increases while the abdominal pressure decreases. And so what will happen is the blood that's in your thoracic cavity will be driven into the heart, and then the blood that's in your common iliac veins, right, will then dump into the abdominal veins there. So again, this will help right, to move. So you have two systems now that we just talked about that help to move, all right, blood, all right, from various areas of your body. The skeletal muscle pump from the lower extremities, and then your respiratory pump will help to move it from the abdominal cavity into the thoracic cavity and then into the heart. All right, so that's the two mechanisms. There's a third one here. So here you can see our respiratory pump. Here's when you're taking your breath, diaphragm flattens, the pressure in the thoracic cavity becomes less than the pressure in the abdominal cavity. So pressure gradient, blood will move from an area of high pressure to low pressure. So it moves into the thoracic cavity. And then when you exhale, the diaphragm then relaxes and goes back to its dome position. Now you've increased the thoracic cavity pressure, decreased the abdominal cavity pressure. So the blood that was in the thoracic cavity vessels, like your vena cava, will push that blood into the heart. And then any blood in your lower extremities will get pushed here into the abdominal cavity. And then when you take your next breath, it moves it onwards. All right, so that's the other mechanism here that helps to move blood back to the heart. Here comes the third one. We talked about venous return. Remember, there's the three here that I talked about. Right? We talked about venous blood pressure here. We talked about skeletal muscle, the respiratory pump. Now we're going to talk about the pressure gradient. I usually save that one for last. Okay. Can we all agree? We know how the pressure gradient works. You guys feel pretty confident about that? Blood moves from an area of high pressure to low pressure. We learned that with the cardiac cycle. That's going to drive this whole thing here. And so that goes back, I'm not going to go back to that slide, with that graph that I showed you, that figure, right? So let's talk about this, right? Systemic gradient. We saw that the pressure in the arteries was at the highest and at the lowest at the veins at the heart. So in the aorta, the blood pressure was the highest, and at the vena cava, it's the lowest, all right? So what do you think? Follow the pressure gradients. So wouldn't you all agree that blood is going to go from an area of high pressure to low pressure? So it's going to try to move it through that whole systemic circuit there. It's going to move from the arteries to the capillaries and then from the veins back to the heart. So that's what this is talking to us about. So when we talk about the blood pressure, we're going to take our mean arterial pressure, our MAP, which what did we say? It was 93 millimeters of mercury. You don't remember? I see some confused looks, right? What's the MAP? Our mean arterial pressure, this one red line here. This is how we figured it out. Okay, we took the diastolic pressure and then we took a third of the pulse pressure, which is the difference between the systolic pressure and the diastolic pressure, which means the mean arterial pressure, pretty much the pressure in the arterial system, right, is going to be about 93 millimeters of mercury, which is still a lot higher than pretty much close to zero in the vena cava. So we can all agree pressure is higher here, lower here, high here, low here. Okay, so as we get further away from the heart, it decreases and decreases. And then we start to return back to the heart. The pressure keeps getting less and less and less and less. All right, keep that concept in mind. All right. So same type of scenario here. Blood pressure in the vena cava is virtually zero, and blood pressure in the arteries is 93 millimeters of mercury. So where does blood want to go? It wants to go from the arteries to the vena cava, to the veins. And then it's going to dump it back into the heart. So this whole thing, that's how we move. That's pretty much the big driving force of blood through your circulatory or vascular system, that pressure gradient. All right, I want you to really kind of keep that in mind. So when you start to exercise, what is one of the things that will happen when you start to exercise? Don't even think about what I just talked about. What happens to you when you start to exercise? Heart rate increases, right? And so therefore your pulse increases. What happens to your blood pressure? Yeah, it increases. Yeah. So all of these things are increasing. All of these things are going to increase your cardiac output. Your heart rate influences your cardiac output. Your stroke volume influences your cardiac output. So all of these things influence the cardiac output. So therefore, all right, when we're talking about our total blood flow, what did I say? Total blood flow and cardiac output are going to be reciprocal. If you increase one, you're going to usually increase the other. So we increase our cardiac output, you're going to increase your total blood flow. Okay? Because when you do that, you're going to increase, and I'm not going to take the time because I know I'm going over a little bit, but when you start to exercise, your mean arterial pressure is going to increase. Look at the equation. Your pulse pressure increases because your blood pressure will increase, right? Your diastolic pressure will increase because, like I said, your blood pressure increases. So that means your mean arterial pressure will also increase. And so if you increase your mean arterial pressure, right? And again, what's our blood pressure in the vena cava? Still 0. So if I go from 93 and I start exercising, let's say I get up to 140 is my mean arterial pressure. I'm not even going to write that. All right. It's just going to look horrible. All right. But now, are we not increasing our pressure gradient difference? Because the vena cava is still 0. But the MAP, the mean arterial pressure, now went from 93 to 140. So increase the pressure gradient, right? That's going to, and what did we say about gradients? The steeper the gradient, we're going to see more movements, more flow. So that's what we're going to see here. So keep that in mind. Let me get rid of all this red. That brings me to that last point down here. Gradient increased by increased cardiac output. All right, so if it works for you to memorize it, just memorize it. All right, let me just see something here. All right, I got to stop. I don't want to, but I have to, because we're going to get into one of